So good morning. Welcome to our third patient and family research Q&A. We're delighted you're here, and it's a very informal session. These sessions are designed for you, our community. And anytime that you have questions, please shout, stop the presenter, put things in the chat, talk amongst yourselves, get to know each other. And we really encourage, if there are questions that you have about research in general, these monthly sessions are all for you. So next month, if there's a topic you want us to cover, please send me an email, send Armel an email, and we will get that arranged for you. They're designed for you with what you need to know. It's not necessarily that we want to teach you certain things. We're responding to what you guys are giving us feedback on. So please let us know. The sessions are recorded just so that our friends around the world can see them at a different time that suits them. So what we normally do is have some personal interaction at the end where we get to know each other, and I'll make sure that that is taken out of the recording so that your personal information is protected. So just bear that in mind if you're asking questions throughout the research slides as well. Any questions before I get started? No? Okay. So let's see if this, I will share my screen. We'll see if the technical challenges continue. As with every good Zoom, you need them, right? Can you guys see that? Yeah, nodding, great. I can see you, but I don't think the chat shows up for me. So just start waving at me if you want me to stop, okay? Right, let's go. So today we wanted to take you back a step and just ensure that some of the information that we've communicated before is really clear, and that we can answer your questions today about some of the processes. So taking us back to the research roadmap, this was developed over an 18-month period. We opened up surveys to our community that were available in multiple languages, asking what their challenges were, what they were worried about in the future. We also held 2 focus group sessions with community members that was facilitated by an independent consultant, and we had amazing representation across Europe and the US. We undertook surveys with our professional advisory board members as well, asking them what their thoughts were on how we progress as a foundation, what they think is available to us, and how they would go about finding treatments and one day a cure. And we did follow-up meetings with them individually, and had extensive, hour and a half meetings with some of our PAB members to really just understand their views and recommendations. We also then had meetings with independent experts from other patient advocacy groups, and that also included institutes like the Milken Institute, and Dr. Christopher Austin was a huge help, and he was previous NCAT's director, which is a group and part of the FDA. And we also had time with Dr. Kakis, who is the CEO of Ultragenyx Pharmaceuticals. So we felt like we had approached this in a really rounded way, ensuring that we have representation from all of our stakeholder groups. I'm just going to press admit and let some more people into the group. There we go. So in terms of our actual research process, as you might know, we have set up an MSF Biorepository, and we've done this through our partner organisation, and they're called Combined Brain. Now, what they do as an organisation, they're a non-profit, and they exist to help other PAG groups like us have some autonomy and an opportunity to take part in research, because traditionally that's something that only might have been realised when you're working with academic institutions. Contract research organisations weren't such a big years ago, so you couldn't do your own. Yeah, Inez?
Can you slow down just a little bit? Oh,
sorry.
Slow down just a little bit.
Sure. So Combined Brain exists to enable groups like us to have autonomy and take part in research. Combined Brain has its own IRB, and that's called an Institutional Review Board, and that is with North Star, and they're an independent US IRB provider. Now, IRBs exist under FDA regulations to ensure that any research you're doing is formally designated to this group, so that they review and monitor the research that you are doing. So it's there to protect the rights and welfare of human research subjects. So we are governed through our relationship with Combined Brain, that they have the IRB in place with North Star. And North Star have a leadership team that has many excellent doctors and scientists on it from institutions like Harvard, the NIH, Brigham and Women's Hospital. So touching on slightly, if you choose to give a sample, what would happen is you would take part in a virtual interview with Combined Brain Biorepository team, and they take you through an extensive interview to ensure that you understand the process, you understand what is happening to your sample, and you know your rights when it comes to your sample. So I just wanted to give you a bit more of an overview about what the Biorepository is and who runs it and who they're governed by. Does anybody have any questions on the Biorepository right now? No? Okay. And anytime, this is super informal, just shout up if I'm...
Okay, what kind of samples is MSF asking to be given through Combined Brains?
Yeah, so Combined Brain can take a wide range of samples. They could take urine, they can take saliva, they can take blood. So depending on your needs or your research project, you would decide which one of those you want to do. And in a second, I'll come on to the couple of projects that we've got running with them. My screen flicks over. Oopa. There we go. So right now, in terms of our research and our sample use, you might remember that we communicated that we are creating iPSCs. iPSCs are induced pluripotent stem cells. And what we have had is two families, one from the 496 variant and one from the 500, who gave blood samples. And from that, the iPSCs are being created, and they are being created by a contract research organization. So that's when we talk about a CRO. They are an independent company, they're a business. You go to them and you say, this is what I would like to achieve, and then they give you back your product. Again, highly regulated, and that works under the IRB system, because remember, you cannot take the sample if you haven't adhered to the IRB process, protocols, and surveys, and ensuring that the sample is meeting all those needs. So iPSCs, just want to reiterate, are blood cells, and they're part of our preclinical toolbox that we took you through that helps us advance our knowledge on Myhre. And those iPSCs might be used in a number of ways, and Mel can talk about that in a second. All of our Myhre patient and control samples, and by control, I mean a healthy sibling or parent, same-sex parent, they are for Myhre syndrome research only. They're not available to other researchers without our consent and approval. They do not go into a biobank that is accessible to others. They have to go through a process of requesting if they would like to use our samples, and then we all have to agree that that is a good use. But an example of where it would make sense for us to share a sample is if one of our collaborating institutions, like Mass General Hospital as an example, or IRB Barcelona, Stanford, or University of Oxford in England, if they received independent funding and had a study that answered some of the questions we had, it would be mutually beneficial for us to give them access to a sample, because there's increased efficiency in time, in budget, and in sample use. Okay, so any questions on that? We can obviously come back to anything as well afterwards. So now our current research projects, and this answers Kathy's question that she had about the MSF conference. So for those coming to the conference, we're providing an opportunity for you to take part in a longitudinal biomarker study. So this is where we will take blood samples at the conference, and we will then track those blood samples every 12 months. Longitudinal just means we're looking at it over a period of time, and that time then gives us the analysis to say, oh, look what's changed, or we're seeing something develop here. So that's what that study is all about. And Mel's going to take you through what biomarkers are in a second in more detail. The iPSCs that we mentioned will be available in August, and potentially will be utilized in our RFP, which was our request for proposals. And that was for our gene and cell therapy project that we have got. So if whoever gets the contract to take forward that work, it might be beneficial for us to give them the iPSCs in that sense. So again, that shows you where you're saving efficiency, time, sample use in that instance. To touch on the RFP in a little bit more detail, we are calling for a team science approach. So this is where we're asking for experts in Myhre to team up with experts in gene and cell therapies to work on a 3-year project and to identify possible treatments and a cure. I really underlined possible because I wanted to just reiterate that research is an investigation. So the purpose of research is that you think you know something, you go in and it might say, yes, you're right. And it might say, no, you're wrong, or it might say something else. Like Amel can speak to this, the amount of research she has done that might have had an unexpected or change, or you have to. So the nature of research is we are doing it to understand. And we're not necessarily saying that we're going to get to X point at this stage. You know, we're learning, we're understanding what Myhre does. And Myhre is a complex and complicated syndrome, as we've talked about in other groups that we've had on here. So the FDA has very, very strict guidelines on how preclinical and clinical work is undertaken. So preclinical is all the investigative side of things, what you do before. It's the work in the lab, it's proving your hypothesis on animal models, it's ensuring that what you are doing meets all the safety standards and protocols and toxicity. And clinical is when you then are ready to take it forward to a clinical trial. But you do not get the opportunity to test anything on a human unless you have met the requirements of the FDA for those safety and toxicity and prove that it's going to hopefully do what you think it's going to do. But having said all that, obviously, the clinical trial is there as a trial in itself, because this is the time that it's being tested in humans. Any questions? I know I've covered a lot in that particular slide. Any questions, or Mel, is there anything you want to jump in and add to that?
Just maybe letting people know what's the FDA, because I'm not sure that everybody knows.
Oh yeah, Food and Drug Association. They're the governing organization for the work we're in terms of drug discovery and putting new drugs out into the market. It has to meet their requirements. Yeah.
Yeah, they are the authorities deciding, in fact, if a clinician is allowed to test a molecule or a drug on humans. And then they are the authorities that estimate if the drug is beneficial for the patient or not. So accepted, approved or not.
Mm-hmm. And this kind of ties into our whole work with the FDA, because we have been liaising with them for the last few months. Zach, who's on the call, is arranging this patient listening session. So your relationship with the FDA starts early, and they want to have a relationship with you to understand the work you're doing, because they are a great organization to also give you guidance on what they're looking for, and ensuring that you are meeting the needs of your community. And that's why we hold these groups. We want to know what you guys need, and we need your feedback, because you are an integral part to the FDA's process of us then getting approval. Okay, I think those are the end of my slides. I will hand over to Amel, but has anybody got any questions now for what we've just discussed in the last few slides?
I have a question. So I was reading about I'm probably gonna mix all the acronyms up and stuff, but the IBCs or whatever you said. And I know, like, one thing that that concerns me is that we have a syndrome that's very much connected to cancer risk. Between the female cancers, and then there's also risk for the some certain tumors coming up in the not the endometrial but the digestive tract, and that's stuff and those areas concern me, because I know, like, when you per se flip the grid or whatever the word, the term is, like flip the script kind of thing. And there is cancer risks that can be associated with over flipping it kind of thing, And so I guess my question is what is the cancer risk? Like, what is the risk of these like doing the biomarkers and I know like that specifically, like taking the samples isn't going to do that, I got that. But when we get to the place where we're like, hey, we're going to trial some things, etc., right? So what's the risk?
These are on. Oh, there we go. Okay, so
Okay.
so Amel, so I guess this touches on cancer risk in general, which we've talked about in terms of the expression and that, but then also how that translates into potential clinical trials and the risks there.
Yeah, that's kind of where I'm at.
Okay.
Yeah.
Thanks.
Yeah. So the just taking your blood to study the biomarkers has no effect on you. It's just
I know, I get that. But when we get to the place where we're at the place where, so I mean, I'm older, so my brain thinks like I'm older. You know what I'm saying? Like, so my brain's like, oh, like what is that? What is we get if we get to the place of we can do something, right? Some treatment things and stuff like that. Like, what is the risk factors? Like, especially around cancer. Especially knowing that we have, our immune systems, and the other part that ties with it is our immune system. So we know from the research from the NIH that our immune systems, some of us over metabolize and some of us under metabolize, right? And we know that, okay? That's very much available research. Some of us over metabolize, vaccines, and some of us don't use them at all. Okay, so those types of things, and when you talk about like the biomarkers and that kind of thing, like in getting into like taking out cells and putting them back in and stuff like that, like I guess I just wonder what are those, because that's where like I'm thinking these are type of stuff that people should know. You know what I'm saying?
Yes, exactly. So that's all the risk. We take that into consideration, and that's the way we choose, in fact, where to do research and which kind of therapy, knowing that there is the risk of cancer. We are not going to do gene therapy, for example, like putting another gene in the patient and increase the number of SMAD4. We are not going to do that because we know that there is the risk of cancer. So all these risks, we take that into consideration, and putting them together, then we can decide which kind of therapy will be without those kind of risk, will not trigger those kind of risk. So thinking about, for example, so the Myhre syndrome, the problem is that there is an error in the gene. So there is a mistake in your gene.
Yes,
so
I know that.
Instead of adding gene therapy like many others disease would do, what we want to do is just remove the mistake, make a correction. Just make a correction so that you will have your normal level of SMAD4. You don't you don't modify anything that could trigger cancer. You just go back to normal SMAD4 sequence. So that's it. So that's the idea. So it's a bit difficult to do because it's way more tricky to make a correction than to just put a gene therapy and put the gene in the patient and trigger everything that can be triggered. So we go for a difficult path, but we are selecting very, very good scientists, the best scientists we can find, and combine with the Myhre experts, all the Myhre, the clinicians that are already working on Myhre for 20 years or more. And we combine those people, the people that are really experts in a technology that would be safe for Myhre patients, and the Myhre experts, and we combine them so that it would be the best team to get to try this therapeutic approach. It doesn't mean that it's going to work, It's just the safest way to address the problem with Myhre.
Anna?
Let me ask you, doctor, when we correct like the SMART4, for example, in this way, for example, Nicole, she has health issues too, but the biggest issue for her nowadays is the autism and cognitive delay, and it's neurologic. It changed too or not? You say the brain is different, the answer in the brain is different than the body, or you don't know?
No. If you correct the gene and you correct it everywhere, then everywhere is corrected. Then your whole body, I mean, not at 100%, but at the good percentage, all the cells are going to be corrected. So if you have a gene editing, like it's on there are a few on the market already, you can put them, if you want to correct the cells in the brain, you can put them in the spinal cord liquids, which is called the CSF, and then it will go to the brain. But the thing is that what we see now in clinical and preclinical phase, it's not yet on the market, is that there is an improvement for autism and cognitive, but you will never get back the whole development that you missed the first years. That's but you still have some improvements. The thing is that we are really at the beginning of gene editing and gene therapy in autism and cognitive delay, cognitive impairment. So we don't have enough years of study back to really have some conclusion. And the way we are going with Myhre, if we go and if it works, it will be in years. So we will have these answers in maybe 3, 4, 5 years when we are going to start with Myhre if it works if it works on animals. Then we in a few years we'll try in patients if we have no toxicity and if all the authorities says it's safe and there is no side effects and the risk is okay, then we'll go. And meanwhile, we'll have the answers from the other diseases. But for now, it's a bit early to tell you. There are some improvements, but
I understood, but I understood. It's amazing too.
So I put this slide just to remind people, maybe people that were not there at the first or the other presentation or talk. I prefer to say talk so that it's really a dialogue between all of us. So that the foundation really advanced now with 4 pillars that are developed at the same time. The preclinical toolbox, that's all the tools we need to study the disease in petri dish or in animals. So that's really what we call preclinical. It means that it's all the study we can do and understand the disease and all the tools we need to before going to human. Then we have the other pillar, which is the clinical readiness. The S is under it, sorry. And that's all, again, all the tools that we need to be able to estimate if a drug is going to be efficient on Myhre patient, if it's going to be safe, if it's going to, how long it's going to work. So all the tools to estimate a drug or a treatment in the patient. And then the third pillar is really the treatment discovery. So the project that we start to accelerate and to focus on some aspect of the disease. And I told you already, so that we are focusing on the gene mutation, so trying to correct this mistake that there is in the DNA. We are focusing also on fibrosis, because that's an issue with of life-threatening issues. In fact, is really linked to fibrosis. And we are helping scientists that are discovering new drugs to really estimate if those drugs are working on patients. The fourth pillar is regulatory interaction with the regulatory and industry So that the regulatory is the FDA, and in Europe, the EMA. So those are really very, very important interactions and collaborations, because they are going to agree or disagree if a treatment is good for Myhre patient or not. So that it's very important that we talk to them, we present them with our tools to estimate the drugs, and our tools to estimate the drugs before, and have this contact with them so that they understand what we want, what the patient needs, what the specifically Myhre patient needs, and they are going to interact with pharmaceutical industry and tell the pharmaceutical industry, this is okay for those patients, this is not relevant, this is not good for the patient, or this is really good for the patient. So it's really important for us to interact also with them so that they really understand what the Myhre patient needs. So so those biomarkers, those biosamples. So a biosample means that it's the small syringe that you see here. A biosamples is when you take something from a human, you take a piece of it. So liquids biosamples is, for example, when you take urine or when you take blood. So it's just taking blood. And in this blood, in this, yeah.
You can just say Anna. I know it's to James.
What? Sorry? Is it?
Right, carry on. I think it was just unmuted. Someone was unmuted.
Ah, okay. So the purpose of taking this blood is to try to identify in the blood what we call liquid biomarkers. So biosamples is the fact to take a little bit of blood. Biomarkers is the things that are in the blood that are different in Myhre patients compared to the large population, the wild type, what we call wild type population. So the example I'll always take is the biomarker for people who have diabetes, for example, is to measure the glucose in their blood. So the glucose is a biomarker. The biosample is the blood. The glucose is the biomarker for the diabetes. If it's too high, if it's too low, it means that you have to modify your insulin or act on your diet. So that's the biomarker, that's the glucose. And what we want to do with this biorepository, so we want to take the blood from the patients, Myhre patients, over 2, 3, 4, 5 years. And we are going to compare what's in the blood, compare to, over time, compared to year 1, year 2, year 3, year 4. That's the longitudinal. And we are going to compare what's in the blood from the patient compared to their siblings or other people that don't have Myhre syndrome. And then from there, we can say, we can take the blood and say, try a molecule, for example, try a therapy, and say, if this molecule that is typical for Myhre syndrome goes back to the level of all the other people, then the treatment works. So that's I'm going to do escape because I don't see you and I don't like that. So that's really the purpose of the biomarker. Do you have any questions?
Yeah, Armel, I see some questions in the chat, so I can just read them aloud, if that's okay.
Yeah, because when I do the presentation, I don't see it's a full screen. So.
Yeah, so one of them is, are there some hypothesis of what biomarkers will be different in controls?
For now, no. So we have, we know a little bit of biomarker in autism in general, but biomarker specific for Myhre, it has never been done. Liquid biomarkers for Myhre patient, we don't know. We will be the first one to do that. Never studied up to now.
Which ones are the ones in autism that have been found?
Well, it depends on the gene, it depends on so many things, and they are not validated clinically. So I'm going to explain you what's the validation. So I'm going to go to the next. Am I still sharing? Yes. I'm going to go to the next.
No, we can kind of see. Oh, there you go. Yeah, you got it.
So the different type of biomarker. So that's the thing that is different in your blood. So we want to have that because we want to monitor. So you have monitoring biomarker so that we can see the evolution of your disease. We have diagnostic biomarker. So it means that if we see that molecule in your blood, it means that you have Myhre syndrome. So that's also very useful. Another type is what we call the pharmacodynamic or response biomarker. So that's really, we try a treatment, and if we see that it goes to a normal level, then we see that it works. Those predictive biomarker safety, that's more for cancer, so I'm not going to talk about that. So why do people want to have liquid biomarkers now? Up to now, when you do a clinical trial, it means that you try a new treatment on patients. So you need to have a way to measure. Is it working? Is it not working? So up to now, it's something very general. You have, do you see a decrease in the stenosis? Do you see can your child walk more in 6 minutes? And it's very variable, and it's very difficult to quantify from one person to another. So the advantage of having a liquid biomarker is it's really quantifiable, which means that you can measure the number of microgram in 1 milliliter of your blood. When you have a blood analysis, you can see that there are numbers really very precisely noted. So that's quantifying, and those are in fact biomarkers that you see on your on your blood analysis. So we can correlate to clinical outcomes. So that means that we can say that if we have that level of that molecules, it means that you are at that level of the disease. So that's extremely important. It's very sensitive and very specific to one type of population. So we are talking about only Myhre patients and not any other patient with autism or any other patient with fibrosis, for example. That's very specific to you. So that if this specificity increases the chance to find the right therapy for you without too much side effects, and really targeted and tailored to one type of patient. So we can, with this analysis of the biomarker, we can also calculate the risk of progression, which is great. And it's cost effective. It means that it's way less expensive to measure something in your blood than to go through an MRI. For example, MRI is thousands of dollars for the clinical trial, while an analysis in your blood will be maybe $100. So it's really cost effective. And it's really personalized management. So we can identify your level 10 years ago was that level. Now you are going 10 years later with that level, and it's your data. So it's really personalized management. So why do we use biomarkers? So I'm talking about clinical trials all the time. I hope you understood that clinical trials, it's when the preclinical, the scientists and the drug discovery people have identified a new way of treating the patients. Then they go to the clinicians, your doctors that you see, and they tell them, we have found something, we have found a treatment, we would like you to test it on your patients. So the clinicians then look at all the proofs. The FDA, the authorities, look at all the proof, and we organize, if it's really good and proven that this therapy can work, then the clinicians and the FDA are going to organize a trial. So a kind of protocol, how we are going to test this new therapy on those patients. And now there was a recent analysis, over 9,000 clinical trials that were done in the past 10 years. And we saw that the clinical trials that were using biomarkers are twofold higher likelihood of approval. So approval, it means that the FDA says, okay, that's a good therapy, it's good for the patients, it's beneficial for the patient, they are going to, we allow you to use these treatments on those patients. So that's the approval. And we have, so the use of biomarkers is really improving the quality of the clinical trials. And usually we have in phase 2 a success rate about 10%. And now we have a 50% phase success when you use liquid biomarkers. So that's really a shift towards the use of, from the FDA, the acceptance. The FDA now pushes the organization to develop biomarkers because they see that it's way more reliable than the old way to do, I mean, the way that is still done, unfortunately, in many diseases, but it's the biomarker is way better. So why is it better? It's we think as a scientist that it's really personalized treatments because with the biomarkers, we can really define a very particular population. So we can design the treatments from one particular population instead of designing treatments for larger population that don't have exactly the same molecules that are modified in their body. So now really, but by being very tailored and accurate to one particular aspect, we get better results. It's also very useful when you have heterogeneity, like we have in Myhre syndrome. We have heterogeneity of the manifestations. So by really studying the biomarkers, the blood of all the different patients, maybe we don't know yet because we didn't do it, maybe we will be able to define smaller population even in the group. I'm not going to talk about basket trials because I think it's for another, when you know a little bit more about clinical trials. So why do we want to start now the biomarkers? We don't have a clinical trial now. We are not going to have a clinical trial even next year. But the thing is that all this study, first it's longitudinal. So we are going to take a minimum of 3 years of blood, and we are going to follow this blood along the 3 years. And then we are going to start to study those proteins that are in the blood. So that's called proteomics study. And this is at least 4 years of study. And then when we have identified, let's hope that we have identified biomarkers, then we need to go through all the process of really evaluating the biomarker in other patients than the one we had, and validate with a kind of clinical study, validate the biomarker so that they can be taken into consideration by the regulatory authorities, meaning the FDA and the EMA.